Hello, now we'll be seeing about week 3 changes in embryology. Week 2 it was bilaminar, week 3 it is trilaminar. Changes similar to week 2, we will discuss the changes um, happening in embryonic area as well as in the trophoblast area. Embryonic area there is change in the shape. In day 14 it is circular, in day 15 it is oval and day 18 it becomes pear shaped having a caudal and a cephalad end. The ectoderm differentiates into surface ectoderm, neuroectoderm, and pluripotent cells. This pluripotent cells forms the primitive node and primitive streak. The primitive streak will give rise to notochord, intraembryonic mesoderm, and primordial germ cells. What is gastrulation? Formation of three germ cell layers from the inner cell mass is called the gastrulation. What are the three layers? Primitive streak, endoderm, and mesoderm. How this primitive streak is formed? Remember the formation of procaudal plate in week 2. What happens is at one particular point the epiblast, the columnar cells are taller than the other area and it is so much attached with the hypoblast. From this area the cells are pluripotent and they tend to migrate downwards between the epiblast and hypoblast and that later becomes the mesoderm cells. Similarly, the cells move migrate along the line and this forms the primitive streak towards the primitive node. You can see the cells, this is the dorsal view of embryo showing the streak. You can see towards the node the cells migrate. So in day 15, the cells from the procaudal plate proliferate and bulge into the amniotic cavity, which elongates along the central axis of the embryonic disc. This is called a primary organizer because it gives rise to notochord and mesoderm. And this determines the craniocaudal axis and the right and left symmetry. It completely disappears on day 26, so it is a transient structure and it marks the beginning of gastrulation. Second, we go to the formation of notochord. If procaudal plate is for cephalocaudal axis, notochord is for ventrodorsal axis. As you can see, the primitive node is otherwise called the Henson's node is the cranial end of the primitive streak. It has many number of cells. And this knot will have a pit called the primitive pit or otherwise called as a blastopore. The cells will become the notochordal process and the cavity inside this pit will become the notochordal canal. And with this blastopore, this cavity communicates with the yolk sac and the amniotic cavity. This notochordal plate will give rise to the vertebra, nucleus pulposus and the apical ligament of Dens. What is neurulation? The ectoderm overlying the notochord thickens to form the neural tube. It extends from the procaudal plate to the primitive knot. And the cranial part becomes the brain and the caudal part becomes the spinal cord. The third germ layer, intraembryonic mesoderm. Remember, mesoderm is also derived from the epiblast. As you can see, between the epiblast and the hypoblast, the cells migrate and come between these two layers. And the one closer to the notochord will become the paraxial mesoderm. Lateral to that is the intermediate mesoderm and very lateral most is the lateral plate mesoderm, which gets differentiated into somatic layer and splanchnic layer having a intraembryonic coelom in between. So this paraxial mesoderm will differentiate into somatomeres and somates. Somatomeres are near the head region and give rise to the muscles of the face and neck and head. Somates are 44 pairs and they are related with the trunk except the occipital one. The occipital one will give rise to the tongue. Intermediate mesoderm will give rise to urogenital system and the lateral plate mesoderm will give rise to splanchnopluric mesoderm, somatopluric mesoderm and the intraembryonic coelom. Connecting stalk is the place where even by week 3 the disc is still bilaminar instead of being trilaminar and that marks the development of a buccopharyngeal membrane and cloacal membrane in the future. Coming to the changes happening in villi, in third week it is tertiary villi. So you can see in this image, uh, initially the core is only cytotrophoblast and syncytotrophoblast. In secondary villi, the mesoderm will invade and it occupies the core. And tertiary villi, there will be capillaries developing inside the mesodermal core. In this image, you can see the brown mesoderm is invading in between the cytotrophoblast, and then inside the mesoderm there is development of the vessels, capillaries, and also you can see the lacunae are beautifully arranged in a radial appearance. This one is the connecting stalk. So to sum up, what is happening? There is a primitive streak and a primitive node and notochordal formation, neurulation, and change in the shape of the embryonic disc. So day 15, primitive streak forms and the embryonic disc is circular in shape. Day 16, it is oval in shape. Day 17, notochordal process will form, and day 18, pear-shaped embryonic disc and development of intraembryonic mesoderm. Day 20, neural plate, neural fold, neural groove forms, and day 21, the first pair of somates will appear. Thank you.